Hi everybody, the title of today's lecture is Early Primate Evolution. This is Anthropology 225 online. The Cenozoic era is divided into a series of epochs, and they're indicated in the following slide. We are currently in the Holocene, and the Holocene's from the present to about 0.01, which is about 10,000 years ago. That's followed by the Pleistocene, the Pliocene, the Miocene, the Oligocene, the Eocene, and the Paleocene. The Pleistocene's from 10,000 years ago to 1.8 million years ago. The Pliocene's from 1.8 million years ago to 5 million years ago. The Miocene is from 5 million years ago to 22 million years ago. The Oligocene is from 22 million years ago to 35 million years ago. The Eocene is from 35 million years ago to 54 million years ago. And the Paleocene is from 54 to 65 million years ago. What I plan on doing is for each of these epochs, excluding the Holocene, I plan on reviewing the major forms of primates that evolved during each of these time periods. So the primates that evolved during each of these various epochs. And we'll start out at 65 million years ago, the very beginning of the Cenozoic. The first epoch of the Cenozoic that I want to mention is the Paleocene. The Paleocene is from 65 to 54 million years ago. It is probably one of the most poorly known epochs. This is not because we don't have a lot of geological samples from this time period, but rather we don't have a lot of non-human primate samples from these geological epoch. Despite the fact that it's one of the most poorly known epochs, it does represent the first radiation of placental mammals. During this time period of the Paleocene, from 65 to 54 million years ago, climates were a lot cooler than they were during the preceding epoch of the Cretaceous during the Mesozoic. We know that climates were cooler based on the flora. We're seeing primarily coniferous trees, trees with pine cones, and many, many fewer tropical plants. Geologically, geologically, the Paleocene was a very, very active epoch in Earth history, and it's marked by the rise of several different mountain groups. It was during this epoch that the Rocky Mountains actually were formed. During this time period, geologically, North America, Europe, and Asia were all connected, so they formed one continental mass. Australia, South America, and Antarctica were also connected, forming a second continental mass. And India and Africa were each separate entities. And India had not yet collided with mainland Asia, so we didn't yet have the Himalayas. So this is just an example of continental position 65, 66 million years ago. Relatively similar to today, but there are some very important differences. Okay, so what about primates? Because that's really what we're interested in. When we're talking about our own evolution and understanding our own evolution, we need to understand primate evolution per se. The earliest primates are known to have evolved from an insectivore-like mammal during this time period, during the Paleocene. This is known, it's impossible to determine exactly how primates are related to other orders of mammals, but there are suggestions, indications from both paleontology, anatomy, and biomolecular studies that primates, tree shrews, and flying lemurs are all more closely related to one another than to other mammals, and thus they are placed together in what we call the superorder Euarchonta. So it's probably the earliest primate probably looks something like a mix of primates, tree shrews, and flying lemurs. And flying lemurs are not related to the lemurs of Madagascar. In addition to these extant mammals, fossil Plesiadapiforms also lie near the origin of primates, either within the order Primates or as another order of closely related archontans. The suborder Plesiadapiform as I noted, constitute a group of primate-like mammals. And these species really flourished during the Paleocene and the Eocene in both North America, Europe, and Asia. So notice these are the earliest primates that we know of, or primate-like mammals that we know of. And they are all from North America, Europe, and Asia, not normally where we consider the evolution of primates to have occurred. The known diversity of these plesiadapiform primates is greater than living strepsirhines or New World monkeys. There are over 50 genera, over 135 species. The primate status of these plesiadapiforms, though, has been questioned for decades, and it's very unclear whether plesiadapiforms are in fact primitive primates or just closely related to the primates. So these are some examples of what some of these earliest plesiadapiforms are believed to have looked alike. So what happened to these plesiadapiforms? Why did they go extinct? Well, there are several explanations for the demise of these primates or primate-like mammals. The first is there was extensive competition with other mammals, such as rodents and early prosimians, so they were basically outcompeted for resources. And also a second hypothesis is climate change, that there was a dramatic drop in sea level during this time, and perhaps the drop in sea level caused some environmental changes that the plesiadapiforms were not able to adapt to. As I noted, their primate status has been challenged for decades. Every decade, the textbooks change as to whether or not plesiadapiforms are or are not primates. So, but for the purposes of this class, we're not going to consider them primates. We're going to consider them primate-like mammals, closely related. The description, what are these mammals like? They have a low flat skull, a long snout, a small brain, large zygomatic arches. And the primary reasons that I do not consider them as primates at present is that they do not possess a postorbital bar. Remember this bony ring surrounding the eye? No plesiadapiform possesses a postorbital bar. In addition, the auditory bulla is not part of the petrosal portion of the temporal bone, another really good synapomorphy for these non-human primates. What are a couple of these ancestral primate features that plesiadapiforms do have that suggest maybe they are in fact primates? Well, they have a nail-bearing hallus in one species called Carpolestes. That is, they have a grooming claw. I'm sorry, they don't have a grooming claw. They have a flat nail on their toe in Carpolestes, and you know that flat nail is a really good primate characteristic. They also have a bulla that, in some species, that is in fact formed from the petrosal, or suggested that they might be. But there are really no direct relationships of relations of Eocene prosimians or later primates to these plesiadapiforms. The most important thing we can get from these plesiadapiforms is a better understanding of the earliest evolution of features that are characteristic of these primates. So, as you can see, there wasn't much that I could tell you for the Paleocene because we really don't have very much about primate evolution during the Paleocene. The second epoch that I want to mention is that of the Eocene. The Eocene is from 54 to 35 million years ago, and it is marked by a major change in fauna. Basically what we see is that all modern mammals start replacing all the archaic ones. So all the archaic forms of mammals go extinct, and the modern ones prevail. In primate evolution, this epoch is marked by the disappearance of plesiadapiforms, so the plesiadapiforms go extinct during the Eocene. We also see the appearance of the first primates that actually represent strepsirhine primates. In fact, the Eocene is often referred to as the epoch of primates of modern aspect, because the primates from the Eocene look remarkably like those of modern-day primates. The faunal changes that occur during the Eocene are tremendous, and it results in changing geography. North America and Europe are increasingly separate and distinct. There are intermittent connections between them, but they're still predominantly separate. There's more regularly connections between North America and Asia. India also begins to collide into Asia, and Africa is still separate from Europe. So just keep these continental positions in mind as we're talking about the evolution and distribution of these primates. Okay? As I noted earlier, we see that the Eocene is marked by the disappearance of plesiadapiforms, as well as the first appearance of primates resembling living strepsirhine primates, living prosimians. So we have lemurs, lorises, and tarsiers all starting to appear during this Eocene epoch. We see a large number of fossils, all generally strepsirhines, from this time period. These fossils are very, very different from the plesiadapiforms of the Paleocene because they specifically have anatomical features characteristic of living primates. They have small infraorbital foramen, they have flat nails, they have grasping hallexes, they have postorbital bars. Good synapomorphic characteristics are found in all these living primates, and this suggests a very different way of life from these plesiadapiforms. Many of the cranial features of these Eocene primates indicate increased reliance on vision rather than olfaction. The postcranial changes suggest an increased importance in manipulative abilities. You need flat nails instead of claws when you're trying to manipulate things with your hands. We also see that they're much more acrobatic and that they have leaping abilities. Okay, so these primates of the Eocene are often referred to as primates of modern aspect, but they're also referred to as crown primates. So here we see no postorbital bar, we see no postorbital closure back here, and we see postorbital closure here. So these primates had a very, very different distribution than the previous epoch. They had a tremendous adaptive radiation. There are 4 times as many strepsirhine genera during the Eocene as there are known today. And there are quite a number of lemurs and Madagascaran primates. And for us to say that there are 4 times as many different species in the fossil record as we see today is tremendous. Now, all these fossil specimens are not equally well known on all these different continents. They're very common in North America and in Europe. There are a couple in Asia, a couple in Africa, but not very many. And they are completely unknown in South America, Antarctica, and Australia. So what are some of the similarities between extant strepsirhines, those are the living prosimians today, and Eocene strepsirhines? Well, basically their mandibular symphysis, remember the 2 halves of the jaw, are unfused in extant strepsirhines. That's also true in the Eocene strepsirhines. They also have an unfused metopic suture. The Eocene strepsirhines have no postorbital closure. They all possess a postorbital bar. They all have nails, no claws. They have forward-facing eyes, an opposable thumb and toe, a grooming claw, a smaller snout, small infraorbital frame, and a small little hole right here, and the cranial blood supply from the internal carotid artery. So we see tremendous similarities between living prosimian primates and Eocene prosimian primates. These primates first appear at the same time in Europe and North America. The fauna, in fact, of these 2 continents are very, very similar. And once again, this was enabled by the geographic connection between these continents at high altitude, as well as high global temperatures, enabling primate dispersion at those high latitudes. There is no clear phyletic ancestor of early primates among closely related mammals. There are two poorly known species from Africa and Asia that may in fact lie close to the origins of all later primates. These are the following: Altanius orlovi and Altanius kulchi. I apologize for my pronunciation. Altanius is from the Eocene in Mongolia, so this is north of Russia, and it's originally described as an early Omomyoid, but it's probably a Plesiadapiform. It has elevated trigonids and tall premolars, and it's considered a basal primate near the common ancestry of the Adapids and the Omomyoids, so very, very early. We also have Kulchi, we'll say, and it's a Paleocene primate from Morocco, which is northern Africa, and it's known only from 10 isolated teeth. It's 50 to 100 grams, so it's relatively larger than some of the other stuff that we've looked at. And it's considered part early Omomyoid, part basal primate, part early anthropoid. It is literally the oldest fossil primate, and it has affinities within the order its affinities within the order Primates are uncertain, but it's most likely a primate. But how it's related to the other primates is not clear. So those are the absolute earliest primates that we know of. After the earliest primates from the late Paleocene and Eocene, we now have these Strepsirhine primates from the Eocene, and they can be divided into two distinct superfamilies. So the Eocene Strepsirhines are divided into two distinct superfamilies, the Adapoids and the Omomyoids. The earliest members of both superfamilies, both the Adapoids and the Omomyoids, are very, very similar, suggesting a divergence just prior to the Eocene, so in the very, very late Paleocene. The members of both superfamilies are Roose Elia, Cantius, and Teilhardina. So let's talk about both the Adapoids and the Omomyoids. These are the superfamily Adapoids. And these guys are lemur-like. They have very large, they're relatively large, comparable to living lemurs in body size. They have a dental formula of 2-1-4-3, so you have 4 premolars, 3 molars, 1 canine, 2 incisors in each quadrant of the mouth. The limbs are similar to modern Strepsirhines, but are more robust. So this is what some of these Adapoid prosimians probably look like, relatively similar to this ringtail lemur here. So how do they resemble living Strepsirhines? Well, they resemble living Strepsirhines in their anterior dentition. To begin with, their lower incisors are small and positioned vertically. Their upper incisors are broad, short, and separated by a median gap. So there's effectively there's a space between your two front teeth. The upper and lower canines are larger than the incisors, and the anterior premolars are caniniform, meaning they're like sharp and pointy instead of square and flat. So what else can I tell you about these Adapoids? Generally, we see that they have relatively long snouts, but they have a small infraorbital foramen, suggesting a reduced reliance on olfaction. They all possess postorbital bars. Their brain case, the size of their brain, is larger than those of the Paleocene forms, but it's smaller than extant lemurs. Their blood supply is from stapedial and promontory branches of the internal carotid artery. And they all have nails, no claws. They also have an opposable big thumb and opposable big toe. So there are 6 families within the Adapids, and I'm going to go through each of them. So we have the family Northarctidae, and it's from the early Middle Eocene of North America, and the best known genus is Cantius. There are numerous species of North Arctidae in North America, and there are 2 of them in Europe. So it's a predominantly North American family. They're all very small to medium-sized prosimians, 1 to 3 kilos. They have a very primitive dental formula of 2143, that 4th premolar again. Their mandibular symphysis is unfused. Their orbit sizes are relatively small, suggesting that they're diurnal. And their postcrania suggests that they are arboreal quadrupeds. The second family that I want to mention are the family Circumonidae, also known as Proto Adapids. These are from the early to middle Eocene. They're predominantly European in their distribution, but there are a couple in North America, Africa, and Asia, not many. They're much more diverse than the North Arctidae. The best known genus of this family is Roose Elia. It's the earliest and most primitive genus. It has a 2143 dental formula, simple tritubercular molars. So tritubercular molars are molars that are almost triangular in shape. They also have a hypocone from the lingual cingulum rather than from the protocone as the North Arctines. The hypocone is one of these little bumps or grooves on your molars, and it's showing where it's coming out of when we make these assessments. So this is North Arctis, and this is Circumonius. This third family is the family Asiadapidae. It's primarily from West India in the early Eocene, basically from the Voston colbines, and it's very probable that it's at the base of the adapoid radiation. They're very closely related to Cephalodapids, and there are 2 genera. In terms of its description, they're about the size of a bush baby, 200 to 600 grams. They have very simple premolars with no nanopithix fold on their upper molars, which is analogous to what we see in bush babies. The hypocones and lingual cingula are weak or absent, and they're very agile quadrupeds. The next family is family Canopithecidae. These are found in Europe, North America, Africa, and Asia, and slightly later in the Eocene. We're looking at mid to late Eocene. Traditionally, they were grouped under the Circumonidae, but now they've been separated out. There are 9 genera and 11 species. The most important one is Margarita stevensi. It's from late Eocene of Texas. And the most intriguing feature of Margarita stevensi is the following. It has a fused mandibular symphysis, which is a clear haplorhine characteristic. And it has an enlarged promontory artery canal, also for larger brains, a good anthropoid haplorhine characteristic. But it has no postorbital closure. So we're seeing a mosaic of characteristics in this species. And then we have another one, same family, is Darwinius macile, which is from Messel in Germany. And we have a young female with almost the entire skeleton preserved, as well as impressions of fur and the remains of even the stomach contents. And she weighed about 660 grams. The teeth and the stomach contents, which contained leaves and fruit, indicate a herbivorous diet. Large orbit size indicates that she was nocturnal. And the long hind limbs with a low intermembral index suggest that she was, in fact, a leaper. This is her. Isn't that cool? Can you imagine finding that? I can't. Very cool. And then we have the family Adapidae. And this is from the middle of the Eocene up through early Oligocene, predominantly found in Europe. In fact, the member of the family Adapidae was the first non-human primate named. It was so unusual that Cuvier didn't recognize it as a primate. They all possessed molarized last premolars. So the premolar, instead of being a tiny little square, is an elongated square. They have well-developed shearing crests and a lot of postcranial peculiarities. One of the best-known species is Adapis paracensis. It's from the late Eocene of France. It has a 2143 dental formula. It has small upper central incisors with a gap between their bases. This is probably for the organ of Jacobson, which is very important for sense of smell, for olfaction. Their upper lateral incisors are very, very small. They have lower incisors and canines, form a single cutting edge, and some people have suggested this is indicative of the development of a tooth comb. There's a lot of controversy about that because there's no wear or hair striations evident on these teeth. We see it has a very long, low skull, flaring zygomatics, a pronounced sagittal and nuchal crest on larger individuals, especially the males. These these little orbits suggest diurnality, and the blood supply is from the internal carotid of the promontory as well as the stapedial. So here's the organ of Jacobson, okay, right here, and just very important for your sense of smell, okay. The last family I want to mention within the Adapoids is the family Cephalodatipid, and it's in the Eocene to late Miocene in Asia. Cephalodatipus nigreri. It's 3 kilos, 2133 dental formula, and it's folivorous. We see that based on sharp crests on its molars and premolars. So this is the first family where we're really seeing a reduction in the number of premolars. So what can we tell based on all this variation in the morphology of these Adapids? What do we really know about their behavior and their origin and their taxonomy? Well, some people have suggested that there's evidence for an ancient African origin for the Strepsirhines. Remember, the earliest ones were found in North America, Europe, and Asia. So an ancient African origin is quite surprising to read about. We see that there are large Adapids from North Africa, such as Kano Pithosids. And we're also in recent years finding an increasing number of tiny primates from North Africa and the Arabian Peninsula, and they have a number of dental similarities to Eocene Adapoids and to Extant Strepsirhines. Just as an example, we have Azebia turki from the Eocene of Algeria, Northern Africa, and has really unusual premolars. The Aviolus suggests this species and another related species, Algirapithecus, possessed a procumbent canine suggestive of a tooth comb. They all have premolars and molar crowns that overlap, as do extant Strepsirhines. So there's some indication for an African origin for Strepsirhine primates, but the majority of it says North America, Europe, and Asia. A big question that results from a lot of this, our knowledge of Adapid morphology and behavior, is some people have wondered, are these Adapidae actually Strepsirhines? Virtually all authors have noted their similarities to living Strepsirhines, in particular to the lemurs. Similarities to the lemurs include their cheek teeth, their configuration of the skull, the postorbital bar, the long snout, nasal region morphology, an inflated auditory bulla, free ectotympanic ring, as well as the carotid circulation. Most individuals within the Adapidae have a stapedial artery of moderate size. So what I want to point out is all these features are primitive features. They retain the primitive condition found in mammals instead of sharing a unique specialization. The Adapids lack the unique specializations of these Eocene primates. They lack the tooth comb, and the tooth comb is probably one of the most clearly distinguishing features of Strepsirhine primates. So, but these Strepsirhine primates that are living today do have some links with these Adapids, which justifies why some people have argued that the Adapids are in fact fossil Strepsirhines. For example, Adapids also have a grooming claw on the second toe of the foot. This has been known from Circomonines and other living Strepsirhines. They have a flaring fibular surface of the talus, a similar arrangement of the cuneiform facets of the funicular. These are features of the feet, and a unique articulation between the ulna and the carpus. So we have, except in North Arctines. So we have a lot of, not a lot, but we have some anatomical similarities between living Adapids, living Strepsirhines, and fossil Adapids is what I'm trying to say. Although living Strepsirhines and fossil Adapids are similar in overall morphology and probably represent sister taxa, there's very little evidence demonstrating a unique phyletic relationship between Strepsirhini and Eocene Adapids. So the answer is probably not. Okay, the second superfamily that I want to mention in today's lecture are the Omomyoids. Okay, we had the Adapoids, now we're going to have the Omomyoids. The Omomyoids are what you might call tarsier-like. They first appear in the early Eocene. They're found both in North America, Europe, and Asia, much like the Adapids. They have a very different evolutionary history, though, in North America and Europe. And this is because in North America, the Omomyoids are very, very diverse. In Europe, there's only one genus from the early Eocene, Tailhardina, and 4 genera from mid to late Eocene. There are dozens in North America. The most primitive of the Omomyoids is Tailhardina, Steinius, and Altaneus, and they're all very, very similar to the early Adapids that I mentioned earlier, such as Roose Elia and Cantius. They're similar in terms of their dental morphology and in the dental formula. They all have a 2-1-4-3 dental formula. Later Omomyoids start to have dental reductions. Okay, so these are some examples of the fossil Omomyoids and current possible Omomyoids. So the family Omomyidae, they basically are characterized by, let's see, did I skip? No. Okay. The family Omomyidae, they have very large procumbent lower central incisors. They have a small lateral incisor, and the canines are relatively small. The premolars in most species are reduced to 3 or fewer, and most have a post or portocingulum, what's called a nanopithix fold, joining the protocone distally. The mandibular symphysis is always unfused in the Omomyidae, and their skull morphology is also very specific. They have a very short, narrow snout. Their palate gets broader as it goes towards the posterior. They have really large eyes. We see both the stapedial and the promontory present, and they tremendously resemble tarsiers and galagos. There's some evidence that the tibia and the fibula are partly fused in some specimens of Omomyidae. We note that the calcaneus is also elongated, and the morphology of the Omomyoid semicircular canals indicates agile or jerky motion, as in extant lemurs, the constant leaping. And so you need your semicircular canals to handle that constant rapid motion. The earliest, it's probable that the earliest and most primitive Omomyoid, Tailhardina, probably gave rise to all 3 subfamilies. The second subfamily is the Anaptomorphinae. These are from North America. They are mostly primitive. They're ancestral to both Omomyoids and the microchords. The earliest genus is Tailhardinia, and found in the early Eocene of Europe and North America. It weighs 50 to 500 grams. They are characterized by having tall, pointed lower 4th premolars and reduced lower 3rd molars. They are probably frugivorous, with a few species showing adaptations for processing invertebrates that have hard shells. There are 2 complete crania remains from the family Anaptomorphinae, and they're known from Titonius homunculus from Wyoming. And Wyoming probably has one of the most detailed records of population and species changes in the entire fossil record. We see remarkable documentation of transitions between paleospecies and a tremendous amount of evidence of considerable parallelism, the simultaneous development of features in different species. This is Titonius. The second subfamily is the Omomyoidae. This is from North America and one specimen from China, so Asian. It's found in the Middle Eocene. We have 100 grams to 2 kilos. They replaced the enaptomorphines in the Middle Eocene. Reasons why probably have to do with global climate change, and they could be derived from enaptomorphine ancestors such as Steinius. The last molar is elongated. One specimen from the family Omomyoids is Coronae vigiensis. It's also from the late Eocene of Texas. It's a nearly complete cranium. It has a body mass of 400 grams. It was probably diurnal based on the small orbits, and it was frugivorous with rounded molar cusps. The third family of the Omomyoids is the family Coronae. This is the European group, and they're found early to late Eocene. There are 7 genera. They vary in size. Pseudoloris is 50 to 100 grams. Microcorus 500 to 1800 grams. So it's very small to small. The upper dentition is 2133. This is characteristic of New World monkeys. The lower dentition has one less tooth. A classic example of a microcorine is Necro Lemur, and we have many complete but albeit crushed crania. We know that they have a very short, narrow snout. They have a bell-shaped palate, a gap between the upper incisors for the organ of Jacobson, large eyes indicative of nocturnality, a large infraorbital foramen. The ectotympanic forms a ring within the bulla, but extends laterally to form a bony tube. And so it resembles prosimians in the position of the ring, but tarsius in the tube. And it also has a partly fused tibia and fibula. So very, very interesting specimen. And so this is Necro Lemur and Pseudoloris. So the tarsids. Numerous fossil primates have been described that are attributable to the same family as the living tarsiers. 3 of them are Tarsius eocaneus, is from the Middle Eocene of China. We have teeth and cranial fragments that are identical to extant Tarsius in orbital and nasal morphology. We have Afrotarsius chatrathi from the Oligocene of Egypt, and we have Tarsius thailandica from the Miocene in Thailand. These tarsid remains appear to document the presence of tarsids in Asia for over 50 million years. So some people refer to the tarsiers as a result as living fossils because these tarsiers look just like these fossils from 50 million years ago. And interestingly, this is very consistent with morphological and biomolecular distinctiveness. So are these Omomyoid tarsiers, or are they haplorhines? Well, the Omomyoids are in fact have a number of features that are in fact similar to tarsiers. They're very small in their size, they have large eyes, they have an elongated calcaneus, and they have a fused tibia fibula. So they have a number of anatomical characteristics. Several authorities actually place the European microcorines within the actual family Tarsidae. Obviously this is debated extensively, and we're not going to take that path today, but I just want you to know that there are features that link them, and some people take it even further. But I want to make it clear that many of the similarities that actually link Omomyoids with tarsiers are very superficial. They're either shared with other European Strepsirhines, or they're actually primitive traits. Eocene Tarsidae fossils suggest that true tarsids are older than most Omomyoids, and therefore Omomyoids are not actually tarsids. Omomyoids, tarsids, and anthropoids all do share a number of features that leads all researchers to group them together in the semi-order Haplorhini, but it doesn't mean that Omomyoids are in fact tarsiers.